Hello everyone, welcome to the new session of Biotechnology in Agriculture. This is the fifth session. Genome editing, also called as gene editing, is a group of technologies that give scientists the ability to change an organism's DNA. Okay, so for a change in the organism's DNA, we follow the genome editing process. What is that? It is a group of technologies that give scientists the ability to change the DNA of a particular organism. Okay, so this technology allows the genetic material to be added, removed, or altered at a particular location in the genome, right? So, in an organism's DNA, if we want to add a part of any foreign DNA, right, we can go through this technology. Not only the addition, we can remove a particular part of a DNA, or we can also alter a particular part of a DNA with this particular technique. Several approaches to the genome editing have been developed. Okay, along the time we are having a development in this particular technique. A well-known one is called as the CRISPR-Cas9, which is a short for the clustered regularly interspaced short palindromic repeats and CRISPR associated with the protein 9, and that is why it is called as CRISPR-Cas9. Genome editing using the clustered regularly interspaced short palindromic repeats. So this is the full form of CRISPR, clustered regularly interspaced short palindromic repeats. So the CRISPR-Cas9 was adapted from a naturally occurring genome editing system that bacteria use as an immune defense. Okay, so this particular technique were naturally found in the bacteria, and bacteria use this as a immune defense, right? When infected with the virus, when the bacteria get infected with the virus, bacteria capture a small piece of virus DNA. So any bacteriophage, suppose this is a bacteria and it is having an infection with the bacteriophage, right? So when the bacteriophage, when attached to the bacteria, this is bacteria, it transferred its genome or the DNA to the bacterial cell, right? So when the bacteria get infected with the virus, bacteria captured a small piece of virus DNA and insert them into their own DNA and take a part of a viral DNA and get inserted into its own DNA, right? In a particular pattern to create a segments known as CRISPR arrays. So that is called as the CRISPR arrays. That is clustered regularly interspaced short palindromic repeats. So like restriction enzymes. So what is the function of the restriction enzyme? Restriction enzyme cut at a specific site on the DNA. Okay, so this technique is a biotechnological adaptation or repurposing of a naturally occurring genome editing system present in the bacteria. So this is the adaptation of a technique that already present in naturally in bacteria, in some of the bacteria. So in the natural context, this system is used to memorize the DNA signature of past viral attack, so that any subsequent viral infection with the matching DNA signature present on the viral DNA can be countered or destroyed effectively. What does this mean? So if any bacteriophage, this is a bacteriophage, get attached to the bacteria, right? And this bacteriophage transferred is DNA into the bacteria. Now this is the bacterial DNA. This is the bacterial DNA. And this is the viral genome or the viral DNA. Viral genome. So what happened? A part of this DNA get inserted somewhere in the bacterial DNA, right? And what happened whenever this bacteria get in future infected with the same virus, right? It can detect this virus and it can make some proteins to get rid of this virus, because the genome is already get inserted into the bacterial DNA. So it's know all the protein that gets synthesized by the virus. So to counter that virus, this bacteria can make certain protein that can destroy this virus. So part of the guide RNA is a complementary to the DNA sequence where the sequence need to be edited. So we require a guide RNA. Okay. So where we required the DNA to be edited, for that we require a guide RNA. Okay. So the other part of the guide RNA attracts and bind to the Cas9 endonuclease. So this Cas9 is a protein, that is Cas9 endonuclease. This guide RNA get attached to this Cas9 endonuclease. Once this whole complex, which complex? The guide RNA and the target DNA and the Cas9. Three things. Guide RNA, that is the first guide RNA. Second is the target DNA. And third is the Cas9 protein. When get assembled, it form a complex. A double strand break is introduced in the DNA by the endonuclease. So there is a cut in the target DNA because there is an endonuclease enzyme. So it can break the DNA at a particular site. So here we can see this is the Cas9, this gray color, that is the Cas9 enzyme. This yellow color is the guide RNA. And here when this guide RNA form a complementary pairing with the DNA at this particular site, the cleavage is done. Okay, the cleavage is done. So the DNA get cut at this particular site. Okay, so the broken DNA undergoes a template based homologous DNA repair where a modified or a edited DNA template is provided that has the desired changes. So here the DNA undergoes a template based homologous DNA repair. Now there is a requirement of repair and insertion of this particular DNA, right? So how this DNA get inserted and the repair of this DNA occur, let us see. As a consequence of such double strand breaks and the template guided DNA repair, the repaired genomic DNA now possesses the edited DNA sequence. So when with the help of some enzyme, this is already, there is a break in the DNA. So it is easy to insert a particular DNA here at this particular site, right? With the help of some enzyme, these two sites get joined. Okay, and now this is a edited DNA sequence. This is the edited DNA sequence because a new sort of DNA get inserted at this particular site, right? So now this DNA is not the previous one, it got edited. So that is why it is called as edited DNA sequence. Now we studied about this CRISPR-Cas9 technology. Okay. Now, what is the significance or the application in the field of agriculture? Right? So the scientists at the University of the California, San Diego have developed the first CRISPR-Cas9 based gene edited plants with the objective of producing drought and disease resistant crops. So this particular technology which occur in bacteria to get defense with the virus now got introduced into the plants so that the plant get a drought and disease resistant crop. So this technology was used in agriculture now. Breaking from the traditional inheritance principle that stipulate that an offsprings acquire the genetic material equally from each parent, that is called as the Mendelian genetics. The new research employs the CRISPR-Cas9 editing to transfer the specific trait from a single parent to the successive generation. So earlier what we are doing, we were totally based on the Mendelian genetics, that is the traditional inheritance technique, right, in which the two parents were taken and the cross between them is made, and we see whether the offspring is having both the desired characters from both the parents in the offspring present or not. So that is by chance, right? But here in this technique, the CRISPR-Cas9 editing, a transfer of the specific trait from a single parent to the successive generation, and this technique is surety of transferring that particular trait from one specific organism to a desired organism in which we want to transfer that particular trait, right? So it has been used to improve the nutritional quality, increase yield, and develop resistance to the various biotic and abiotic stress in several crops. So this CRISPR-Cas9 has been used to improve the nutritional quality of the crop plants, increase the yield of the crop plants, develop resistance to the various biotic and the abiotic stress in crops, including rice, tomato, and soya. As we know that the traditional method take years to develop a different traits, that is the CRISPR-Cas9 is comparatively a rapid than other genetic manipulation techniques. So this technique is very different from the other technique because it takes very less time to manipulate the DNA, and we get the desired trait in a specific organism or the organism in which we want to incorporate a particular gene or a trait. Furthermore, the CRISPR-Cas9 technology is relatively simple. It is a simple technique, precise, and can be used to create many beneficial traits in plant as it target multiple genes. However, the revolution brought by the CRISPR-Cas9 in the plant's molecular biology has raised various concerns for its off-target effects. So there are some off-target effects also, okay, and at times low efficiency. So if we see the application of the CRISPR-Cas9, that is of a greater aspect than if we see the deleterious effect or the low efficiency in some of the plants, okay. In addition, there are regulatory issues and several challenges for its safety and use in the future. Efforts are being undertaken to improve its on-target efficiency and several modification of the Cas9 enzyme. So keeping this in mind, we are doing the modification on the Cas9 enzyme so that other regulatory issues can be in our control, okay. So our work carried out, it is a preliminary stage and there is still more to be done. So scientists are working on this challenges so that we can have a total, there should not be any disadvantages of the CRISPR-Cas9 use, okay. So many scientists are working on this, and in future we can have only the significance or the beneficial effect of the CRISPR-9 and no deleterious or harmful effect of using CRISPR-Cas9. Okay, so here we are having some few genetically modified plants and their acquired characters. So here in the table we are having the crop, name of the trait, gene that has been transferred, that is mentioned in this particular table, and application, that is in the third table. So here we are having some stress resistant genetically modified crops. What are those? Arabidopsis and tobacco. So name of the trait or the gene transfer, that is the mannitol. That trait helps the plant, that is the Arabidopsis and tobacco, helps the plants to withstand high saline condition, high salt condition, and enhance germination rates and increase the biomass of these two particular plants. Rice. Embryogenesis, abundant protein gene from the barley. So this is the particular gene transferred for this particular trait, more tolerant to the drought and salinity than the conventional varieties of the rice. So if the embryogenesis, abundant protein gene from the barley has been transferred to the rice, okay, from barley, this particular gene is transferred to the rice. Now rice is more tolerant to drought and salinity if we compare to the conventional variety of the rice. Next crop is soya bean. HAHB4 transcription factor gene from the sunflower plant. So this particular transcription factor gene has been transferred to the soya bean. And what is its effect? HB4 soya bean named eco-soy, tolerant to drought and herbicides. Wheat. HAHB4 transcription factor from gene from the sunflower plant. So same gene has been transferred to the wheat also. Again, the wheat is now named as eco wheat and has increased the yield up to 20%, and also this wheat is drought tolerant. Next, genetically modified crops for enhance the paper quality. Arabidopsis. Gene encoding an Arabidopsis thaliana protein regulated in the transgenic plant with up to 45% less lignin. So if the lignin is less, okay, then it enhances the quality of the paper. Again, Arabidopsis. Gene encoding an Arabidopsis thaliana protein 20% more wood than the conventional variety, and it is ready for harvest in 5.5 years instead of 7. So we can save our time as well as 20% wood than the conventional variety, so we can have more paper. Genetically modified crop for improved product. The first crop is potato. Gene involved in starch synthesis. Okay, 60% more starch than the non-genetically modified plant, which makes the potato take up less than fat during frying, resulting in a lower fat product. So we are having lower fat potato plant, okay, and that is 60% more starch. So this potato is favored and that is genetically modified. Second is the maize and soybean. 2 stack genes confer the tolerance to the herbicide glyphosate and 2,4-D-choline. So these are the two herbicide which has been used in the crops in the field of maize and soybean. So that if this particular gene has been transferred to the maize and soybean, then they are having tolerant to these herbicides. Third is the apple. Gene involved in non-browning. Apple named Arctic Gala had non-browning traits. So sometimes the apple that get brown when cut or exposed to the air. So if we transfer a gene involved in non-browning, so that particular apple do not get brown, okay, and that is the name of that apple is Arctic Gala. Next is the rice GR1, that is the vitamin A. The rice, a gene for the vitamin A has been transferred to the rice GR1. Maize phytoene synthesis gene and gene from the bacterium Erwinia uridivoya Golden rice, this is very, very important, is an effective source of vitamin A in humans. Now we are having a variety that is called as the golden rice. It also provides you vitamin A along with the starch. Biofortified rice, iron and folate. Two genes from the daffodil, that is the Narcissus poeticus, a bacterium that is called as Erwinia iridioria, affect the carotenoid biosynthesis in the rice. So now this biofortified rice with improved iron and folate, with improved mineral bioavailability and with high content to essential amino acids. So two things has been introduced in this biofortified rice, that is iron and folate, right? So this is all about this session. We will meet with one more interesting session. Till then, thank you very much.